am going to start, and people are still coming in, as you can see, so it'll take me a little bit to go through my introduction. So I'm Kathy Giacomini, so please welcome to Lightning Talks Room 3. These talks are designed to feature research and regulatory science. And remember, at the very beginning of this whole show, I showed how FDA is funding CERSIs throughout the countries, Centers of Excellence in Regulatory Science and Innovation. I'm going to be moderating the talks today. The way this session is going to work is that we will have 3 back-to-back lightning talks, 5 minutes. And the speakers will be available on a panel for the final probably 10 minutes. So please submit your questions in the Q&A. There's a Q&A function, and then I'll read them at the end and during the panel discussion. I'm now going to introduce all 3 speakers now, and then they can begin speaking in turn. So the first speaker will be Florence Bourgeois from Harvard Medical School. She's going to be talking about pediatric drug policy, so that will be an interesting topic. The second speaker will be Gita Thanarajasingham from Mayo Clinic. She'll be talking about wearables and patient-reported outcomes in chemotherapy. And the final speaker will be UCSF's Leslie Wilson, who will speak about patient preference in islet cell transplants. So let me begin with Florence. And Florence, I'll try to have a timer and ring you when there's 5 minutes up.
Terrific. Thank you for the introduction. Can you see my slides?
I do.
Wonderful. Thank you. So my name is Florence Bourgeois. Thank you for having me today. And I'm delighted to speak to you today on policies advancing drug development specifically for children. So to set the stage, I want to start by orienting everyone to an important feature of pediatric research, which is that there's a lot less of it compared to adult research. So as this figure nicely shows, beginning in the 1970s, there was a rapid uptick in the use of clinical trials to assess interventions and in particular drugs in patient populations. But pediatric trials have consistently lagged behind. There are a number of understandable reasons for this, which we won't get into today. But in terms of what this means for clinical care, it's that in many cases, we don't have all the evidence that we would necessarily like to inform our use of drugs and other interventions. For medications in particular, it's estimated that about 52% of drugs do not have pediatric prescribing guidelines. And so what we do as physicians is we extrapolate findings from adult studies, use the product in kids, looking without necessarily having age-specific information on dosing, safety, or efficacy. In terms of the scope of this, it's quite widely, it's a fairly wide problem. So approximately 85% of hospitalized children are treated with an off-label medication, and the majority of pediatricians knowingly prescribe off-label drugs. So this is in part related to how policies regulating drugs in the US have been developed and implemented. Starting in the 1960s, the FDA began to require that both safety and efficacy information be available in order to approve a drug, but children were not enumerated in any of these policies until 2002. And that's when the Best Pharmaceuticals for Children Act was passed, followed by the Pediatric Research Equity Act, or PREA, in 2003. So these are the 2 principal programs governing pediatric research at the FDA. There are a number of other factors as well, but these 2 are the main ones that I'll focus on today. And specifically, what I have been interested in my group is to assess the impact of these legislations in order to understand what are the actual increases in pediatric drug research that have occurred under these programs, what are increases in pediatric drug labeling, which is how we can offset off-label prescribing, and importantly, what can we say about where these pediatric programs might still fall short and how can they be strengthened? We've used a number of different types of databases to do these, some of them publicly available, some of them proprietary. We've also had freedom of information requests. We've collaborated with the European Medicines Agency and the EU as well to understand their policies. And this line of research culminated in a systematic review that was published just a few months ago, where we assessed all of the pediatric studies that were requested or required by the FDA under these 2 programs. This encompassed all drugs and biologics approved by the FDA from 2002 through 2018, so since the inception of these programs. And the FDA during this time had issued a total of 389 pediatric study requests for 274 new drugs and biologics, and this represented 320 indications. So these 320 indications are the ones that the FDA felt were relevant to kids and for which they either requested or required that sponsors conduct some type of pediatric trial so that we can inform our use of these agents in children. And these were the principal results of this review. So on the x-axis, looking at years after FDA approval for an indication, you can see that at 5 years, both for PRIA and BPCA, there's a relatively small proportion of studies that have been completed. So for PRIA, about 23%, and for BPCA, 13%. And importantly, the time to completion of these trials was over 4 years. What does this mean for the clinical use of these agents? Almost 2/3 of these drugs and biologics were available on the market for at least 5 years without any pediatric safety or efficacy labeling information. So again, what these programs are trying to achieve ideally is to reduce that time window so that we can have less off-label drug use. This work also informed a policy analysis that was also recently published. And here we put forth 2 main recommendations around these pediatric programs. The first was that policies could be modified to ensure prioritization and study of drugs that are most relevant and of highest priority to pediatric patients. So if we can't study all these drugs, given that these hundreds of indications may be too many to accommodate in pediatric trials, can we identify the ones that are most important? And secondly, these initiatives must aim to increase the number of studies that are conducted before or soon after drug approval in order to ensure timely availability of high quality prescribing data to reduce that off-label window. Thank you very much.
I'm Gita Thanarajasingham. I'm a lymphoma hematologist at the Mayo Clinic in Minnesota. I'm also a health outcomes researcher focused on evaluating the tolerability of cancer treatment to adult patients. Thanks for the opportunity to present our project on quantifying physical function in cancer patients on behalf of our Yale Mayo CERSI team in collaboration with the FDA Oncology Center for Excellence. I'm a clinician first, so I'd like to start with a clinical scenario. This case is actually inspired by the experience of the father of my collaborator at Yale, Dr. Kerry Gross, who wrote a very thought-provoking piece in the Washington Post about his dad's cancer treatment experience. So the patient is an 80-year-old man with relapsed Hodgkin lymphoma, and we start him on an antibody drug conjugate. The drug is known to cause an initially mild but progressive peripheral neuropathy or damage to the nerves that can become severe in some patients. And this patient lives independently. He manages all of his own ADLs, and he learns about this side effect and asks, how is this going to affect my ability to golf, garden, drive? And do I, as his clinician, have the evidence to answer his questions about physical function? He's not asking about safety, but rather about the tolerability of treatment. Our cancer clinical trials have historically emphasized response and overall survival rates, but we don't do as good of a job evaluating how well our therapies improve cancer-related symptoms, the burden of side effects, or how they impact a patient's physical function, all factors related to tolerability. The FDA OCE has done excellent work on measuring symptoms and function in cancer patients. They've defined a core set of outcomes, including symptomatic AEs, disease-related symptoms, and the one that we'll focus on here, which is physical function, defined as the ability to carry out day-to-day activities that require physical effort. Physical function can be measured in a variety of different ways, including from the perspective of the clinician, using scales like the ECOG performance status. Secondly, from patient-reported outcomes with patients responding to questionnaires about their physical functioning. Third, using performance tests like the 6-minute walk test or the timed get up and go. And fourth, using digital health technologies like wearable devices. We're collaborating with the OCE to investigate the rigorous measurement of physical function by these 4 modalities. We plan to evaluate 2 populations of adult cancer patients undergoing first-line cytotoxic chemotherapy, breast cancer and lymphoma patients. The experience of patients with solid tumors and those with hematologic malignancies can be very different. So this is a particular strength of the study. Another strength to highlight is the innovative approach. We're going to use a wearable device, a Fitbit in this case, as well as a unique application called Hugo, which is a patient-centered data sharing platform. The Hugo platform will allow us to aggregate real-world EHR data with wearable and ePro data to better characterize symptom burden and functional status, and be able to tie it back to clinical outcomes such as acute care usage among these patients. It's patient-friendly, so the same platform that can deliver and record the PROs retrieves patients' electronic health data from multiple sources. The aims of the study really are to firstly see how do we do measuring physical function using the 4 modalities. Secondly, we want to see how these data track together and what defines meaningful change. Do we see a decline in steps or a drop in the moderate to vigorous physical activity, MVPA, when a patient reports a decrement in their physical function? How do these data change over time on treatment? Do they predict clinical outcomes, such as dose reductions or hospitalizations? And lastly, given the unique approach and platform, we want to take the opportunity to understand what patients thought of it. So we'll assess the burden and usability of the pros, the wearable, and Hugo from the participants conducting an exit interview. So here's our schema. Essentially, we're going to enroll patients, provide them with the Fitbit, and get the Hugo app on their mobile devices. And through the course of their follow-up, we'll collect data representing the 4 modalities of assessment of physical function over time. After 12 months on study, we'll conduct an exit interview and the patients will enter follow-up. After initial consent and enrollment, patients can be followed remotely if they're not treated at our sites, which is really pragmatic. So ultimately, there's an increasing recognition that accurate measurement of how patients feel and function on treatment, tolerability, in addition to safety, matters from a regulatory as well as a clinical standpoint, and it matters to patients. We have to evaluate the physical functioning of cancer patients on studies, defining the right metrics, how to implement them, how to analyze the data, and how to interpret these results. It's an important step forward in patient-centered regulatory science. We can understand how to use digital devices and PROs to design meaningful, potentially comparative tolerability endpoints in oncology studies. And I'm optimistic that this work and many ongoing efforts on cancer treatment tolerability will improve the experience of patients like Dr. Gross's father and the many cancer patients I treat. Thank you so much for the opportunity to present. I'm going to turn it over to the next speaker.
I'm Leslie Wilson, and I'm going to talk today about the use of stated preference for islet cell transplant. And the FDA has really shown in a lot of ways that it's committed to capturing the patient voice and applying that voice to regulatory decisions. And related to this, CBER, the biologics group, has sponsored 3 stated preference studies to develop examples to inform the benefit risk assessments. And I'll talk today about one of those, the hard to control type 1 diabetes study. 70,000 of the 1.6 million people with type 1 diabetes in the United States have hard to control diabetes. And this is characterized by high glucose variability, hypoglycemic unawareness, and a high mortality rate of 20 to 50% at a fairly young age, 27 to 45 years. And current treatments are insulin pumps with continuous glucose monitoring devices, but it's expected that islet cell transplant will come up again for review in the next year. Islet cell transplant is when insulin producing cells are removed from the donor pancreas and transferred into the diabetic through infusions into the liver portal vein. And then the beta cells will begin making insulin. So the question is, ICT has not been approved in US yet, despite being approved in Europe for quite a while and in other countries as well. So how do the benefits outweigh the risks? And our objective is to get the patient perspective on how they weigh the risks and benefits as a help to the FDA in their decision-making coming up. Islet cell transplant has met its research targets in 80 to 90% of the cases, still has some safety issues, mainly the ability to isolate the pancreatic islet cells, need to reduce the need for immunosuppressants, and the benefits are reducing the hypoglycemic episodes for different amounts of durations, and what patients care most about is achieving insulin independence. So we sampled 90 adult patients with hard to control type 1 diabetes around the US and constructed this discrete choice measure to measure how they weigh the risks and benefits. And on one side of the slide, you see an example of what the survey looks like. The attributes of risk and benefit are on the left, and then there are different levels. And you ask the patients, if these were your only options, which would you choose, option 1 or option 2? And then you repeat this 18 times, asking for different levels for their choices. Then we do a regression to get preference scores, and we do a latent class analysis to demonstrate preference phenotypes. And here's just a slide which will show you, so you can read it, the attributes of risk and benefit. And essentially, these are risks or benefits, chance of achieving clinical treatment success, and the duration of that success, extent of insulin independence, which isn't the main outcome, but what patients care about most, expected reductions in the risk of long-term complications, unproven, but still important, and then the benefits or the risks, adverse effects of the treatment, long-term serious complications, including bleeding and rare deaths, and restrictions due to the immunosuppressants, and then time and support that's required because often you have to have more than one islet cell procedure. And our results are that essentially that patients are still risk averse, but they are willing to trade the risks for maximum benefits. And you can see this by the coefficients here, where you see that for the attribute of serious complications, it has the strongest preference, which is a negative preference at 15% risk of 2.03. But you also see from the other preference scores that they are willing to trade like 5 years of insulin independence at 1.75 for a 5% risk of serious complications. So we also showed that two phenotypes emerge. One is a risk-averse group, and the other is a benefit-seeking group. And the easiest way to show you this is to look under the chance of serious complications under the risk-averse group. You see that their coefficient is now negative 6.55 instead of a negative 2. So they're very risk-averse. Whereas in the benefit-seeking group, if you look at the insulin independence, you see that they also have a higher 6.54 coefficient, showing their strong preference for the benefits. So in conclusion, we have high risk type 1 diabetes patients. They want to avoid the high risks of islet cell transplant. However, they are willing to make those trades, and they really care most about insulin independence. CBER believes that this preference information will be really helpful to add to the risk-benefit assessments that are anticipated for approval that's going to come up this year. We also showed there are two phenotypes of patients, risk avoiders and benefit seekers. And this is important because this information can encourage approval for subsets of patient groups.
Okay, great. All three of you. That was fabulous. We had a very nice, you were very clear. So now I'm opening it up for question and answers, and I see one from the audience. And that one, I believe, is for Florence. How can we improve industry adherence to completing required pediatric studies? How does this differ in various devices?
Thank you for that great question. That's terrific. That really gets at the heart of much of the overriding question for the work we've done. So in terms of how can we improve things, it's important to understand that the FDA really has only so much authority to compel any sponsor to conduct or to initiate and conduct a pediatric trial. So I would say that it's important that we understand the incentives, what's important to sponsors, and what would more meaningfully and effectively incentivize the initiation and conduct of pediatric studies. And some of that might be that there's a more tiered approach. So you produce a certain amount of evidence, you get part of the incentive, as opposed to this one-off approach we have that's decoupled from the timing. So right now, sponsors are eventually choosing to do these trials, but as I showed, it can take years before they do so. So can we tweak that so that they need to be incentivized by earlier conduct of the trials? Another consideration is that Congress could give the FDA additional authority to require pediatric trials, and could even give them post-approval enforcement tools, which could be monetary penalties, or it could even bring into play sort of the approval of their adult products, which is really what they care about. So making that in some aspect of that contingent on the fulfillment of required pediatric trials.
Great, thank you. I have a question here. It looks like it's for Gita. Are there particular medical devices that you are lacking to accompany your clinical trials?
Good question. So kind of thinking about adult cancer trials, there's lots of opportunity for medical devices. When I think in the sphere of assessing tolerability, which really centers around incorporating more patient-centered assessments, I think things that are out there that are being developed that have become more popular, especially in the last 5 years, are patient-centered platforms like Hugo. We don't want to have, patients having to do paper surveys that someone's transcribing. Ideally, we'd like it to be a same platform that sort of integrates their, health information, their PRO data, shows it to them in a patient-friendly format, and has the opportunity to make it such that they can share it with their clinicians as well, given some of Ethan Bosch's work on sort of real world use of PROs. So I think that's a great opportunity. There's been no one dominant platform or sort of device that incorporates wearable data and can do this. You see that we're using sort of two approaches in our study, but I think that's a great opportunity for innovation among our colleagues, both in industry and academia.
Okay, Gita, you have one other question. Let's take that quickly. And then I have one for Leslie also. Are patients required to wear their Fitbit certain parts of the day? Will certain patients take off their watch during certain activities?
Really good question, Alan. So I feel like I should just invite you to our sort of protocol development meeting that's happening, because this is one of the questions that has come up. We're actually doing sort of a cross-sectional review of other studies that have used Fitbit to try to be as standardized as we can with what's in the literature. And I'll tell you right now that there's not that much standardization. Obviously, we are going to give the patients an opportunity to take off the Fitbit. But, as we're defining which metrics we want to use, some of these metrics may be related to sleep and other things. So I don't think we'll have them take it off the entire time that they're sleeping. So basically, to be determined. I'll get back to you on that.
Okay, great. This question is for Leslie. Okay, this is going up to 10,000 feet. Can you comment now on how patient preference is being used and incorporated in the FDA in their approval process? And can you provide any examples for how it's actually being used?
Yes, it is being used, but it's new to them in these meetings where they're evaluating the clinical risk and benefit. They also allow assessment of risks and benefits across patient preference as well. There's been one main example of approval of a weight loss device, a surgical weight loss device, where they have, where it didn't meet safety criteria. I mean, it met safety criteria, but it didn't meet its main outcome. But the patient preference information was so strong that they did approve it. And then, there are other examples of getting to become more familiar with patient preference as a type of data by having more meetings with the people that are making decisions, clinical risk benefit decisions, and allowing them to become more familiar with that information as well. So there's a lot of back and forth going where preference researchers are learning about what's acceptable in the measurement of preference, as well as the reverse.
Great. All right. I want to thank all three of you. This is a lightning session, and these were lightning talks, and they were great. So thank you all. And I think we're having a 10-minute break before we return to the major session. So thank all of you.